हेलो। सिक्रीशन के बारे में बात करेंगे। एक्स्ट्रा सेलुलर सिक्रीशन जो होता है वो मेन फंक्शन होता है सेल ऑर्गेनेल में गोल्जी कॉम्प्लेक्स का। तो द स्टडी ऑफ सेल सिक्रीशन इन रिलेशन टू ईआर एंड गोल्जी सिस्टम इज जस्टिफाइड एट दिस पॉइंट बिकॉज दिस ऑर्गेनेल आर वंस मोर डायरेक्टली इन्वॉल्व इन द सिंथेसिस, transport and release of macromolecules from the cell. The relationship between Golgi complex and cell secretion was postulated by Kazel in 1943 in his study of goblet cells. Cell secretion is not confined to animal cells alone. In fact, plant cells secrete polysaccharide and protein to make their soluble. Furthermore, the enzyme present in lysosome was produced by a kind of secretory process. The secretion process in pancreas 6 consecutive steps. In a glandular exocrine cell, such as that of pancreas, the following 6 steps that involve the endomembrane system may be recognized. Step 1. The ribosomal stage. The initial stage involves the synthesis of protein in contact with the polysomes attached to the RER. This step, which is actually begin in the cytosol, was studied in chapter 8 protein synthesis में आपने ये पढ़ा है। इसमें ये chapter 8 की बात कर रहा है इस बुक का। तो दूसरा होता है step 2 और the cisternal stage. This correspond to the vectorial transport of जो पॉलीसोम अटैच्ड होते हैं। थोड़ा सा बता दूं आपको, आरईआर जो होती है उस पर राइबोसोम जो अटैच होते हैं तो उनको पॉलीसोम्स कहते हैं। उसमें प्रोसेसिंग होती है प्रोटीन की, जो mRNA लेके आता है न्यूक्लियस से साइटोप्लाज्म में। आपको मालूम है प्रोटीन सिंथेसिस में आप पढ़ चुके हैं। तो द सिस्टर्नल स्टेज क्या होती है? दिस करस्पॉन्ड टू द वेक्टोरल ट्रांसपोर्ट ऑफ सिंथेसाइज्ड प्रोटीन्स इनटू द सिस्टर्नी ऑफ द ईआर and was also, and the student should bear in mind here the mechanism of binding of ribosome to the microsome membrane, the signal hypothesis with the signal codons in the mRNA, the signal peptide, the ribosomal receptor proteins, the signal peptidase, and the mechanism by which the protein is processed and stored within the ER. ये आपके सिलेबस में है नहीं स्पेशली मेंशंस, लेकिन ये आपको प्रोटीन सिंथेसिस तो आना ही चाहिए। तो आपको अगर नहीं कैच कर पा रहे हैं, ये वैसे प्रोसेस होता है कि ईआर ल्यूमेन में जो होता है प्रोसेसिंग होती है, राइबोसोम पे सिंथेसाइज होता है प्रोटीन, तो ये उसके बारे में बात कर रहा है। तो उसके बाद प्रोटीन जो होता है एक मैक्रोमोलिक्यूल है और ईआर में वो आता है इंट्रासिस्टर्नल ग्रेन्यूल की फॉर्म में। तो इंट्रासेलुलर जो ट्रांसपोर्ट होता है, इंट्रासेलुलर का मतलब आपको पता ही है, विद इन द सेल जो ट्रांसपोर्ट होता है। तो सिक्रेटरी प्रोटीन्स जो होते हैं, जिन प्रोटीन्स का सिक्रेशन करना है, वो आरईआर के थ्रू उनको ट्रांसपोर्ट किया जाता है। ट्रांसपोर्टेड थ्रू द आरईआर एंटर द सो कॉल्ड ट्रांजिशनल एलिमेंट्स विच आर लोकेटेड एट द बाउंड्री बिटवीन द ईआर एंड द गोल्जी कॉम्प्लेक्स. These transitional components have ribosome attached to most of their surface except in the region facing the cis or forming phase of the Golgi, which are smooth and bud off into vesicles similar to Golgi peripheral vesicles. According to this study of Pallade and co-workers, this vesicle reach the next station of the secretory pathway and fuse with large condensing vacuoles of Golgi that are present at maturing phase. The intracellular transport of the secretory proteins have been followed cytochemically by the radio-labeled precursors such as 3H leucine, either injected into animal और जो होता है रेडियोएक्टिव जो ल्यूसिन होता है उससे इसको फॉलो किया जा सकता है। Slices of pancreas are exposed first to a leucine-free medium, which produces depletion of leucine endogenous pool. After a short time, the tissue is submitted to a medium containing radioactive leucine for a minute, and this process is then followed by the chase, which consists of washing and incubation with a solution containing excess non-labeled leucine. It is possible to observe in fixed tissue. Isotope is localized in the ER of the basal region. Newly synthesized protein processed then into Golgi complex. It becomes progressively concentrated into this region and pro-zymogen granules or condensing vacuole surrounded by a membrane. After a longer time, the label is found principally in the zymogen granule in the lumen of the acinus. Since the intracellular transport की जो रेडियोटोग्राफी की स्टडी हुई है, द रेडियो एक्टिविटी जो होती है वो जो होती है गोल्जी कॉम्प्लेक्स में क्या होती है, कंसंट्रेशन होती जाती है. तो पैरोटिड ग्लैंड में इसका अध्ययन किया गया है. तो वेव लाइक मूवमेंट ऑफ जो पल्स लेवल्ड सिक्रेटरी प्रोटीन होता है वो इंट्रासेलुलर जो होता है कंपार्टमेंट, उसमें RER गोल्जी कॉम्प्लेक्स, इमेच्योर ग्रेन्यूल्स, मैच्योर सेक्रेटरी जाइमोजन ग्रेन्यूल्स, इस तरह से ट्रेवल करता है. Since the intracellular transport is measured against an apparent concentration gradient, it is to interest to determine what, if possible, metabolic requirements are. The transport of the newly synthesized polypeptide chain from ribosome into the cisternae of the ER does not require additional energy, seems to become controlled mainly by structural relationship of the large ribosomal subunit with the membrane of the reticulum. When the process of protein synthesis is inhibited by puromycin, the incomplete peptides found are transported into vacuolar cavity. Transport from ER to conducting vacuole is blocked by inhibitor of cell respiration or oxidative phosphorylation. इससे ये कंक्लूजन निकलता है कि गोल्जी कॉम्प्लेक्स में जो ट्रांजिशनल एलिमेंट बनता है ER और स्मॉल वेसिकल गोल्जी कॉम्प्लेक्स, वो एनर्जी डिपेंडेंट लॉक होता है इन द ट्रांसपोर्ट. सच ए लॉक में रेगुलेट द फ्लो ऑफ सेक्रेटरी प्रोटीन्स. और एनर्जेटिक्स की जो लेटेस्ट स्टडी है उसमें क्या होता है कि जो गोल्जी ऑर्गेनेल होता है वो हाई कंसंट्रेशन ऑफ NADPase होता है और वो प्रोटॉन पंप की तरह कार्य करता है. स्टेप 4 जो तो क्या होता है, स्टेप 4 जो होती है कंसंट्रेशन ऑफ सिक्रेटरी प्रोटीन होती है. ड्यूरिंग दिस पीरियड the condensing vacuoles are converted into zymogen granule by the progressive filling and concentration of their content, and finally acquire their characteristic electron opaque content. This conversion is not dependent on a supply of metabolic energy, since it continues after inhibition of glycolysis and respiration. Step 5 होती है intracellular stage. The previous step culminate with the storage of secretory products into secretory granules, which are released when the appropriate stimulus, a hormone or neurotransmitter, acts on the cell. This storage provides a mechanism by which the cell may cope with the demand for secretory products that exceeds the rate of synthesis. This mechanism of storage is utilized not only in protein secretory cells but also other producing smaller molecules such as peptides and amine. Step 6 होती है exocytosis. The discharge of the secretory granule is affected by the process of exocytosis, which involves its movement toward the apical region of the cell and fusion between its membrane and the luminal plasma membrane. As a result of the fusion of membranes and the consequent fission with elimination of the layers in between, an orifice is made through which the secretory product is discharged. The mechanism of exocytosis was first described in the adrenal medulla after electric stimulation of the splanchnic nerve. Biochemically, exocytosis requires the intracellular elevation of calcium and production of ATP. Thus, exocytosis is an energy-dependent process and signifies the second energy-requiring step in the entire secretory process. Presumably, part of this energy requirement is related to the process of membrane fusion-fission taking place during exocytosis. But is it possible that energy could be consumed partly in the propulsion of granule to the apex? The process of exocytosis involves the presence of protein of 47,000 Dalton called synexin. Synapsin, which could regulate the calcium-dependent molecular events involved in the membrane fusion and release of catecholamine. In the parotid gland stimulated by adrenergic drugs during acting on beta receptors, the zymogen granules are discharged by hexocytosis in a sequential manner in the order in which they present at the apical region. It has been postulated that cyclic T5-AMP may be implicated in the process of exocytosis. In fact, the secretion can be induced directly by the use of dibutyryl cyclic AMP. A pancreatic cell in which protein synthesis has been inhibited can go through an entire secretory cycle of transport, concentration, storage, and release. This fact suggests that within a period of time of one cycle, the synthesis of new membrane proteins serving as containers of the secretion it is not required. In other words, intracellular membranes have much longer half-life and are utilized extensively during this secretory process. The membrane of the zymogen granules may be regarded as vacuole that shuttles between the Golgi apparatus and cell surface. In the removal or excess membrane from the apical region of the cell, it has been suggested that patches of membrane are invaginated from the surface as small vesicles that move back into the Golgi region to be utilized in the packing of more secretion. Thus, the process of exocytosis is coupled with that of endocytosis. The specificity of this recycling process appears to depend on the formation of coated pits and vesicles which separate the membrane patches from plasma membrane by a mechanism of selective endocytosis. This is the way the extracellular secretion takes place in the Golgi.